Hello, my name's Mike Burrows. I'm a professor here at SAMS, and one of my real research interests is the effects of climate change on marine organisms. And I'm giving this lecture as a guest lecture on the H3 Marine Conservation module. Normally I like to give this in person, but under these circumstances, I'm going to record my presentation today for all of you. In this talk, I'm going to be talking about the impacts of climate change on the ocean and the marine life that lives in it. I'll take you through the threats that climate change poses in terms of trends in temperature at global scales and local scales, looking at the intensity and duration and frequency of extreme temperature events known as marine heat waves. We've been doing quite a lot of research at that at an international level over the last few years, and the research findings are really interesting in terms of understanding what's going to happen in a warming climate. And then I'll take you through some examples of responses of organisms to climate change and developing broader expectations for how those organisms ought to respond, both at a local scale and across their geographical distribution. So we'll be talking about changes in abundance of species, introducing the concepts of perhaps a species response curve in terms of how a species might change with temperature in terms of its abundance, say, or physiological capacity to operate. Be talking about shifts in distribution patterns, and actually then taking a side step into looking at this metric called the velocity of climate change, which has proved really useful for producing first order estimates of how much species should shift under climate change, with real implications for conservation. And then I'll touch on the concept of reshuffling of biological communities. When some species are declining, some species are increasing, distributions are changing, cold water species are moving out of warmer areas and warm water species are moving in. We're creating new communities, and the reshuffling of those communities can be measured in particular ways. And I'll introduce the idea of thermal affinities for different species, and kind of touching a little bit on how new interactions and altered ecosystem functions might be occurring. We all know now what the global trend in temperatures is looking like. After a few years where it was debated whether there was a hiatus going on, it's clear that since at least the beginning of the 20th century, there's been a fairly uniform increase in temperature. And certainly since 1970, across the globe, land and ocean, here's a data set from the US National Oceanographic Atmospheric Administration, showing that at least since the 1940s, there's been nearly a degree and a half change in temperature. If we look at the globe over more recent times, so from 1980 to 2015, here's a map of the trend in temperature in 1 degree grid squares from a monthly data set of sea surface temperature from the Hadley Centre. And you can see that large parts of the globe have been warming up to 0.5 degrees centigrade per year, and some places faster than that. Although not everywhere in the world has warmed. In the Southern Ocean, south of the polar front, it looks like over this period at least there's been a degree of cooling. There are various explanations as to why different parts of the world should be warming while other places are cooling, and I'll not go into that here, but in the Southern Ocean, greater surface mixing is thought to be bringing cooler water from deeper layers to the surface. So while the global trend is uniformly upwards, there's considerable variation from place to place, and there's also those patterns are not uniform over shorter periods. So for example, from the early 80s through to the end of the 20th century, the North Atlantic increased in temperature uniformly, whereas since 2000 there's been a degree of apparent cooling in the North Atlantic from 2000 to 2015. Changes and trends vary from place to place, and this is quite important in understanding and explaining why not everything we see in the ocean, not all the changes we see are consistent with a steady, uniform increase in temperature. Here's some pictures from maps of the trends in temperature over those same periods for the UK. And the reason I'm showing this is because that my interests in trying to observe locally the effects of warming climate on our coastal marine species since 2000 have been slightly frustrated by the fact that during most of the period over which I've personally been collecting data, there's been a steady, well, a kind of not a warming period basically, so a slight cooling from 2000 to 2015, which I'll show you later in some data I've collected. Yeah, it's kind of makes it harder to kind of immediately see the effects at a local scale, and this kind of variability in place to place from trends will continue into the future, but the upward trend in temperatures will be evident at a global scale. So where I would like to start is by looking at how individual species might be expected to change in abundance at any one particular place, kind of looking at the local response to changing temperature, either in terms of a trend in temperature or an extreme event. And I think there are big lessons to be learned from the biogeography of a species, essentially where the species lives in terms of temperature on the globe, and where within that range of temperatures that the species is experiencing does the marine protected area or the particular location that we're interested in fall. Lessons to be learned from geography. And here's my particular interest in the intertidal, around just behind the back beach by the castle. I've been looking at changes on this shore here for at least the last 15 years now, I think, to try to see if we can see at a local scale what's going on. And there are a couple of species here which I do anticipate seeing changes in relation to climate. You can see this shore is more or less vertical, but in this photograph it was mostly dominated by barnacles, and there's a lot more seaweed there now. But the barnacles I'm interested in live at the very top of this band of barnacles, and they're a warm water species. And here's a photograph of those warm water species. I like to include a scale object here, a penny, which we all know how big that is. And here there are two kinds of barnacles. There's also some mussels in this picture, but there are two kinds of barnacles here. One of which is an Arctic species, or boreal if you like, and that's a species called Semibalanus balanoides. It's fast growing, lives lower down on the shore than the other species I'm going to talk about in a minute, and grows to full size within a year or two at most. So these diamond-shaped barnacles here are Semibalanus balanoides. There's another kind of barnacle here which is browner in colour, and it's got a more oval opening, and that's a genus called Chthamalus, and the species here is Chthamalus stellatus. There's also another kind of Chthamalus species here, just to confuse you, called Chthamalus montaguei. And the critical thing about the Chthamalus species is that they are warm water species, so their distribution extends from a northern limit in northern Scotland and Shetland, down to the coast of North Africa, even as far south as Senegal, we think, for Chlamydia stellatus. So you've got here a pair of species, one of which is a cold water species, one of which is a warm water species, and they're existing together in the same place, but the temperature is changing. The warm water species in Britain extends basically up the west coast. It's completely absent from the east coast of England, although since this map was made in 1939, we do know that the warm water species has now extended down as far as Aberdeen and even the coast of Fife in very small numbers. So this is a species at the very edge of its distribution range. So it's kind of, if temperatures decline, it's likely that this species will retreat, and if temperatures increase, this warm water Chlamydia species is likely, or the two of them are likely to advance in their distribution and certainly change in abundance. To understand what's happening when temperatures increase, it's important to think about the physiological performance of the organisms across the ranges of temperatures that they live in. Here's a plot from a paper by Hans Portner in Science in 2008, showing the scope for aerobic performance across the temperatures that an organism is normally found in. Hans Portner's got the idea that the ability to deliver oxygen to your tissues at different temperatures is what plays a major role in determining how species can operate at different temperatures. So that essentially at the extremes of the temperature range for this particular species, the organism is no longer able to deliver enough oxygen to its tissues to allow the species to persist. So typically, aerobic performance or physiological performance in general tends to increase with increasing temperature up to an optimum, a peak, if you like, in the middle here. I'm going to attempt to draw something. Yeah, so there's a peak around here at which physiological performance is at a maximum. And then as temperatures increase beyond that maximum, then there's a decrease in performance, slowly and then increasingly more rapidly towards a point at which the species can no longer deliver enough oxygen to its tissues. So what's called anaerobiosis starts. So life can continue without oxygen, but obviously only for a very short time, anaerobically. And then ultimately the temperatures are so warm that enzymes and proteins within the organism start to so-called denature. They'll irreversibly change into a form, and effectively that's the point at which an organism dies. We see the same sort of pattern if we look at the average abundance of species at particular parts of their distribution range. This is a plot from a recent analysis I published in Nature Climate Change in 2019. If we look across a whole range of species and express the location in which the abundance is measured relative to lower and upper limits, here the lower limit is the 10th percentile of the distribution range of the species, and the upper limit is the 90th percentile of the distribution range of temperatures. We see that essentially things get more abundant, or tend to get more abundant, towards the center of their distribution range. It might be offset slightly from that, but essentially as the temperature deviates from that optimum temperature, which might be physiological, so abundance declines. So you'll see what happens. Consider what happens in terms of warming then. If we can divide that species range into a cold half and a warm half, increases in temperatures for species in the cold half of their distribution range, which is the blue part of this plot, will produce an increase in abundance. So climate change will benefit those species in the colder part of their distribution range. if those species on the warm part of their distribution range, and then that's that descending part of the graph here, then an increase in temperature will take that species further away from its physiological or ecological optimum. So this is a relatively straightforward way to think of things, but it's very powerful, and all it demands is that we know something about the thermal responses of a particular organism across a range of temperatures, and also where we are when we're looking at that species to anticipate the effects of its response to climate. Increasing in abundance when the species is in the colder parts of its distribution range, and decreasing in abundance when it's in the warmer part of its distribution range. So what's happened around the UK since the late 19th century? Well, it's pretty much the same as the global pattern, except there's a lot more noise around that pattern, and less of an obvious straight line increase, I would say. You see periods like the 1960s and 70s where there was a reasonably coolish period, and then a rapid switch beyond there to the late 1990s, and a kind of a hovering around kind of a half a degree increase since then. I mean, I think you can see that where the trend is likely to go, but it, again, as I said earlier, you get lots of year-to-year variability. Okay, back to those barnacles, and here's histograms which express the distribution of that species across its geographical range. So for the cold water species, those bars express the number of coastal places where this species is actually found within its geographical range. So semi-valence Balanoides typically is found between 0 and, let's say, 15 degrees, although it's some extension of its distribution into warmer places. And that means that we can express the place where the centre of its distribution range is around 7 degrees centigrade. For Cathamlus stellatus, the warm water barnacle, the centre of its distribution range is probably around about 18 degrees centigrade for the average annual sea surface temperature, which is somewhere down in Spain or Portugal. But 7 degrees is more likely in kind of northern Norway. Southwest Britain, where I'm about to tell you about some changes that have been observed, lies more or less in the middle of these two plots, but for semi-valence Balanoides, it's towards the warm end of its distribution range, whereas in Cathamlus stellatus, it's towards the cool end of its distribution range. So I think you can kind of get a feel as to what might happen over a time. And so since the 1950s, people have been counting these things in Southwest Britain, and indeed the changes in these two species reflect those changing temperatures. Here's a plot of the ratio of the warm water species to the total number of barnacles on a seashore near Plymouth, related to the temperature anomaly in a particular year. So each dot on this plot represents that ratio of the number of warm water species to the total number of barnacles. And what it means is that effectively, as in warm years, the warm water species dominates, and in cold years, the cold water species dominates. So that shows how relatively quickly changes can happen in these particular kinds of species. Short-lived, but very sensitive to temperature in these places. You can even see the two species being distributed across warm gradients, temperature gradients on a very, very local scale. Here's a rock near Millport, and on the southern flank of this rock, which which is exposed to the sunshine, the southern species is dominating, and the northern flank of this rock is dominated by the northern species. And you can see this locally around the lab. So I'm really interested in understanding what the temperatures experienced by those individual barnacles actually are. And here's an in-situ temperature data logger, of which you'll find several out on the back shore here. It records temperatures every hour and stores about 2,000 temperatures. So that keeps going for about 2 or 3 months. And these records have been going back now 15 years. And what they show you is the body temperature of the organism, the operative temperature. People have developed more sophisticated versions of that particular logger, embedding the logging equipment inside muscle shells to make robo-mussels and inside limpet shells to make robo-limpets. But what they do show is that, and very surprisingly, is that on the high shore, on very warm days, often in early summer, temperatures can exceed the lethal thresholds for these two species. Semibars barnoides is killed above about 44 degrees centigrade, and Thamylus needs a temperature of nearer 50 degrees centigrade to kill it. So you can see in over this period here, the cold water species was experiencing lethal temperatures on at least 3 days out of that period. And shots of the barnacles before and after show that those cold water species did indeed die over that period. So many of the effects of climate warming may be evident very quickly during extreme temperature events. Looking over the number of years that I've been collecting this data, we can see that in some years there are lethal temperature events, but not every year. So this particular pair of species can persist because we get larval settlement and growth of new organisms in the years in between the lethal temperature events. If there was a lethal temperature event every year, it's unlikely that we'd see more than one species in this place. Just the warm water species would prevail. And that kind of thing might explain to a large extent why the distribution ranges of the two species are what they are. In a recent review, came out last year in Nature Climate Change, I was part of a larger group which looked at the reported effects of marine heat waves across a whole range of different, mainly coastal marine systems. This is a collaboration between ecologists and physical oceanographers. Physical oceanographers looked at the incidence of heat waves using these very long-term, high-resolution records, and us ecologists reviewed the literature and analysed the responses that we see. And what we did see, so a heat wave is defined as, we define the heat waves as a number of days above a certain threshold temperature, which is, in our case, we decided that we needed 5 consecutive days above the 90th percentile of the expected temperature for that particular day. And here's a plot using my temperature data logger data with the red line expressing the 90th percentile of temperature for that particular day of the year. And here in 2018, we see we had early on a couple of periods of warm weather in which, for at least 3 weeks, the average sea surface temperature exceeded the 90th percentile, and you can see a reference for that. What this study showed, and we were able to show quite nicely, was that the number of days that any spot on the globe spends in the heat wave state has been increasing with temperature, as you might expect. And across the globe, some parts of the globe are seeing a much greater increase in heat wave states over this time. Some of the effects has been seen. Well, the most dramatic and the most famous is the effects on corals. The phenomenon of bleaching happens when heat waves happen. So, the longer the heat waves, the more intense the coral bleaching. Seagrasses also decline when heat waves occur, as do giant kelps off California. As part of our analysis, we looked at where the reported effect of the heat wave had occurred in relation to the geographical range of each species in the survey. And what was very clear was that heat waves had a much more negative impact on species if those species were close to the warm edge of their geographical ranges. Nearly 90% of the effects of heat waves were negative at those warm edge locations. Towards the cold edge of the species distribution range, the balance between positive and the negative effects of heat waves was pretty even, so 50% positive, 50% negative. Another important outcome of recent work is that it's clear that the effects of temperature extremes and trends in temperature can be strongly mitigated by local effects. So on land, it's clear that the amount of shading a slope gets, and the amount of over canopy cover in forests, it means that those places underneath those, in those shady places and under the canopies, may be protected from the effects of climate change. So when looking at changes at a local scale, you have to be aware of a potential modification of the trend because of some local effect. And we can see that on our rocky seashores, in as much as the sheltered places where there are thick, luscious canopies of brown algae like Ascophyllum, egg-rack, and the other fucoids, tend to have a greater proportion of cold water species than those places in more wave-exposed conditions where there is no protection from the heat in the sun. And the more warm water species occur in wave exposure. So like forests on land, macroalgae can protect cold water species from the effects of heat waves. Okay, so that was all about how abundance changes in terms of trends and short-term events in response to climate change. A really important effect of climate change is just the overall shift in species distribution ranges. So over a long enough period, we'll see a movement towards the poles of species that presently live in warm, temperate and tropical environments. So those species are moving, they're shifting their distributions to stay in the same temperature ranges as the temperatures go up across the globe. So the lines of equal temperature, or isotherms, tend to move away from the equator towards the poles. So distribution shifts are a main response to warming temperatures. Here's those barnacles I keep talking about, and roughly the range limits align with the 7-degree February sea surface temperature isotherm. So as those isotherms change, we would expect the distribution limits of these two species to also change if temperature was indeed limiting their distribution. And there's some evidence for that happening. In the English Channel, this purple volcano barnacle, Perforatus perforatus, extended its geographical range about 120 kilometres eastwards up the English Channel at a rate of about 33 kilometres per decade, from around about the Isle of Wight in the early 60s up to the early 60s to now as far east as Hastings in the late 90s. Here's another warm water species, the purple topshell Steromphala umbilicalis, whose polewood range edge is in Orkney. There seems to be some evidence that the populations of this particular species in Orkney are increasing and starting to appear in places where they previously hadn't been seen. So potentially a range edge extension is going on here at the same time. Here's another one we might consider watching, that barnacle Semibalas balanoides again, extended all the way up as far as the average position of the pack ice in the 1950s. Now that's changed a lot since then, so a re-analysis, or we would expect this species to have extended its northern limit far further north since the 50s. So how do we develop expectations for how far species should shift to track changes in temperature? Well, we can look at these lines of equal temperature, or isotherms, and the rate at which those isotherms are shifting over time. If temperatures limit the edges of the distribution ranges of the species, so if those isotherms shift, so the species should shift with them. If they're able to colonize new places which are newly warm enough at the leading edge of the distribution range, and they're being removed by extreme events or an inability to cope at the warm edge of the distribution range. And there is a metric which allows us to get a real handle on how fast distributions should be shifting, and it's called the velocity of climate change, and it's really just the ratio of the local trend in temperature over time to the spatial gradient in temperature over time. So it gives a metric which is in kilometers per year. It has a direction which is the same direction as the spatial gradient, and it gives a direct expectation for how far things should shift under climate change. So you can see in this lower map here, the red areas are where the spatial gradients are intense, and the blue areas are where the spatial gradients are relatively shallow. And the top part is the warming rate, so those areas warming a lot are in orange, those areas not warming at all are in blue. So there's a lot of spatial variability in both the trend in temperature and in the gradient in temperature. That means we're going to anticipate seeing big shifts in distribution ranges of species in some places, but not others, from the velocity of climate change. So essentially, when the velocity of climate change is fast, we will likely see rapid shifts in distribution ranges. Velocity of climate is fast when spatial gradients are very shallow. That means, so over time, if say you're at a particular temperature and the overall temperature increases, you're going to want to go, you'll have to go a very long way to find that same temperature in the future. Velocity is also fast where the trend in temperature is high, and conversely, where spatial gradients are relatively sharp, where you've got cold water next to warm water and there's an increase in temperature, there isn't much distance to go to find a cold enough place if everything's warmed up by the same amount. We can get negative velocities of climate where oceans have cooled, and that indicates expected shifts towards warmer places, and not many places in the world show that. The median over the last 50 years velocity of climate has been about 20 kilometers per decade. So on average, over 50 years, 100 kilometer shift in ocean isotherms towards the poles. On a local scale, you can start to see some of the differences we might see on the west coast of Scotland. Warming's been less, and the isotherms, which are these white lines here, are closer together. In the North Sea, the isotherms are much further apart, and there's been more warming. And the result of that is that over the past 50 years, the velocity of climate change on the west coast is only around 2 kilometers per year, or 20 kilometers a decade, whereas in the east and the North Sea, we're getting places where the velocity of climate is 20 kilometers a year or 200 kilometers a decade. Really big shifts in the North Sea. And there's some evidence from fish species that kind of distribution shift in, for example, cod has been happening. A test of this is just to kind of compare the reported shifts in distribution with the shifts predicted from isotherms, and essentially the answer is yes. When you've got long-term observations of ocean species, there is generally an agreement between velocity predictions and distribution shifts. This is probably the most reliable comparison of distribution shifts with velocity by Malin-Pinsky in 2013 in Science, and he showed that basically, yeah, the taxons shift their distribution to match the local shift in isotherms. So all this has got quite a lot of importance for conservation. I think it begins to be obvious when you're considering particularly changes in distribution, how marine protected areas, which are essentially static, fixed areas that have been designated for their particular conservation value, may not be protecting those species into the future. There's the possibility that species will disappear from those marine protected areas, even though there's good conservation action to try and reduce the impacts on those species. So we need to consider the likely time that species will remain in those marine protected areas, and we can get a handle on that by thinking on where those species are within their geographical distribution inside the MPAs. Climate residence time is an important thing, so that those places where the velocity of climate is lower, then we anticipate holding on to the distributions of species for longer in those places. Might be a good kind of place to consider as a sort of refuge from the impacts of climate change. Knowing where the species actually is itself within its temperature range is really important. So range edge populations of important species at the trailing of a warm edge, probably not a good place to put your MPA. And alternatively, if it were possible, shifting designations might help to kind of overcome this effect of the overall poleward shifts of species. And there are alternatives too. So non-spatial measures for conservation may well be, because they're applied more broadly, may well be more effective in terms of protecting a species from the effects of climate change. So we need large-scale kind of efforts potentially wide scale. So what specifically might we start to think about in terms of protecting marine species under climate change? And here's a result of a little paper that I recommend that you find, and considering what different sorts of features one might consider for protection under climate. So running through these, the idea that migration corridors might be needed to allow species that are changing their distributions passage through to new thermally suitable habitats. Maybe it will also be worth focusing efforts on biogeographic boundaries where breaks in habitats might prevent poleward extension. So if we want to protect that species, we better look after it in the last place that it could possibly be. And as I was saying earlier, that species are likely to be increasing in abundance and expanding in terms of the range. If they're to be protected somewhere and for the long term, potentially those parts of the habitat that are the cold edge of their distribution range are the places where at least climate change is unlikely to be the major impact on that species in the coming decades. So if we can find parts of that species range which, well, the coldest parts of their ranges may well be the places to look after these species for the long term. Less likely is finding places where temperatures have increased less. I mean, there is variability in the rate of temperature increase, but as we can see, oftentimes that variability is not persistent from one decade to the next. So somewhere that's not cooled much in the last 20 years is unlikely to be somewhere that isn't going to warm in the future. So the past heating is no guide to what's going to happen in the future. And finally, probably worth thinking about where there is most genetic variability in a species. So oftentimes a leading edge populations may have less genetic diversity than trailing edge, just parts of the distribution range. And I think we need to give some careful thought to how that genetic diversity is going to be preserved into the future, even potentially translocating populations to preserve that diversity. In Scotland, in 2011, lots of interest was the process of designating areas involved all sorts of different kinds of considerations, and one of them was this least damaged, most natural designation, in which those least used areas of the coastline were seen as most natural and potentially places to conserve undamaged resources. I mean, I think that really lets some of the more damaging activities off the hook in some ways. But fortunately, the designation of coastal MPAs at least doesn't seem necessarily to directly reflect that least damaged, more natural consideration. Okay, here's a paper from. Here's a graphic from that paper I was talking to you earlier about, just to show graphically some of those points about where best to put your marine protected areas. If the top left here, if your reserve is towards the trailing edge of the distribution and the species range is moving with climate velocity, with that big arrow there, then you can see that little square is very quickly going to be lost outside the distribution range, and so that those reserves will be rendered useless fairly quickly. Whereas if at the leading edge of the distribution range, there's a lot longer for climate to change before the the reserve exits from the bottom there. And a neat, if it were possible, probably requiring international cooperation, is to set up essentially a chain of reserves, and those might ensure continued protection. Okay, the last bit of the talk, I'm going to be just outlining some work I've done very recently on measuring ecological responses to climate change. I think climate change is a very long and slow process, but we do need to know how we're progressing in terms of particularly community composition. If you've got a whole range of species, what's happening on average across all those species in terms of response to climate? And what I'm going to be talking about is kind of the thermal affinities of a whole bunch of species, and indeed trying to work out what the average thermal affinity across those species is, and seeing whether that thermal affinity is changing in relation to climate change. If we get a shift from cold water to warm water species over time, that shows us that the composition of the community is changing, and that the services, the ecosystem services that community delivers to the ecosystem and to people that live in coastal areas may well be altering because of that alteration in species composition. Here, those barnacles again. The cold water species is these green polygons here, and the warm water species is the warm polygons. So matching that with average temperatures gives that histogram that I showed you earlier. And if we could do this for a whole bunch of species, here's all the species that live on UK rocky shores, we can start to get a real picture of the kind of the mix of cold water and warm water species across the community, and the relative fate of those communities, those individual species, is going to let us know how that community is faring. And to do that, we're going to use the community temperature index, or have used the community temperature index, and that is basically the average thermal affinity of all those species by taking an individual species, assigning it a temperature, and then getting the weighted average of that temperature for each species, where the weighting is the presence or the abundance of that species. And we can see how across space the composition of communities changes, and rocky shores from warm water species in the southwest and west of Ireland to cold water species across the east coast. So that the communities are changing as temperatures are changing. Plants seem to be responding slightly different from animals. It's more of a north-south shift, whereas it's more of a northeast southwest split for animals. Might be because perhaps plants and animals are controlled by different seasonal temperatures. Might be. So if we can get that average index, we can plot how the community itself has changed over time. And here I bring you back to one of the issues I was talking about earlier, the fact that since we've started looking at temperatures, the responses of communities to changing temperatures in Britain, there's actually been a slight downward tip, but it's likely to go back up again. In southwest England, the slight decline in average thermal affinity is happened across the board. In Shetland, however, we have the longer term data series, and then there we do see a steady increase in all the species and all the animals in terms of their thermal affinities. Plants seem to stay the same, likely because those plants have very similar thermal affinities, and they're near the middle of their distribution range, so they're neither declining nor increasing. But what's critical, and what you should take away from this as a wrap up, is that in terms of climate change, really there's only one thing you really need to know. Where you are defines whether where the species is within its range defines whether that species can be considered as a warm water species or a cold water species. If you're in a location where the species is generally found in colder places than yourself, then that's a cold water species. And then if you're in a place where, if you've got a species which is generally from a warmer place than you presently reside, they're a warm water species. And with an increase in temperature, what we call warm water species will increase and what we call cold water species will decline. Take that away and then you can more or less extrapolate to every problem that you might face trying to conserve marine species under climate change. Briefly, at the very end here, this is you can stop here if you want to, but this is just a larger global analysis of patterns of change in fishery species and plankton across the North Atlantic. I took data from a whole range of different fishery surveys from North America and Europe, and from the continuous plankton recorder from the SAMS Hardy Foundation for Ocean Science. So in Plymouth, the continuous plankton recorder survey. And I wanted to do the same things I did with the rocky shore species over a 30-year period. And like rocky shore species, you can see that essentially wherever you look, the composition of communities follow the local temperatures, both in fishery species in the top and plankton in the bottom. And if you look at trends over time, then you can see in some places clear shifts from cold water species towards warm water species, here expressed by that community temperature index anomaly here, expressing just basically the difference between an annual value and the long-term mean for that particular box. This IBTS here stands for International Bottom Trawl Survey, and 56 north, 10 degrees east, somewhere in the North Sea. And again, the general pattern everywhere is that warm water species tend to increase and that cold water species tend to decline. So if the thermal affinity of the species is for colder temperatures than you presently reside in, and then you're currently looking at, then those species either don't change or don't increase, but warm water species are definitely increasing.